Hello, everyone. Thanks for joining us today for our e-seminar series, weekly seminar series on translational biomedical engineering. This is the 91st e-seminar that we started in June 2020 after the pandemic. So I think I hope that you appreciate this e-seminar series. So today we are hosting, it's a great pleasure, and we are very happy to host Professor Marco Bonizzato, if I pronounce his last name. Yeah, so that's good.
Very good.
He's an assistant professor at Polytechnique of Montreal, and also is affiliated at Department of Neuroscience at University of Montreal, and he's also co-founder of Neural Drive company. Is a startup company. And then I'll introduce him later more. Next week, so if you have any question about his talk, just put your question in the Q&A box, and then the chat is for informal kind of greetings and chats, and you can introduce yourself and talk to your flow audience. So next week we give a little bit of break to our e-seminar. We don't have e-seminar next week, but for getting the most updated information of the upcoming seminar series, you can always follow our Twitter account with the handle of TransLBME, and also myself and Dr. Akbari on LinkedIn. So we post our upcoming e-seminar series, and these seminars is sponsored by Montreal Transmedtech Institute, that they actually started sponsoring this almost from the beginning of this seminar series. By that, I will introduce our guest, Professor Bonizzato is a control and electrical life science engineer with over 10 years experience in implantable brain-computer interface and neuromodulation technology. He has a unique double expertise in a neural prostheses and machine intelligence and optimization. He is an assistant professor of electrical engineering at Polytechnique Montreal and adjunct professor of neuroscience at University of Montreal. He's directing the sciNeurotech lab. The research goal is developing the entire translational arc of new neurostimulation therapies aiming at restoring sensory motor function after neurotrauma, from discovery in rodent to application in human medical technology, tailored and personalized to each user by artificial intelligence. So he has been publishing in great journals. These are things that I added because he's very humble. He has been publishing in Nature Family journals and high impact journals, and he was very active in entrepreneurship. As I said, he's a co-founder of Neural Drive. So by that, I asked him to share his screen and start his presentation. Thank you so much.
Thank you very much, Suman, for the kind presentation. And in a second, I should be able to share the slides. Here we are. Okay.
All good. All good. Yes.
Okay, thank you. So we're going to talk today on neuromodulation to restore movement. And neuromodulation in particular is delivering electrical stimulation to the nervous system so that you can modulate the activity of certain networks within the system. That means basically delivering very small currents to targeted locations in the brain, in the spinal cord, in the nerves. You might have already seen some of the applications. In particular, I studied paralysis, so the lack of movement, and how through neuromodulation we can recover better movement function, or even like spontaneous recovery of control of movement, so the capacity to return to a higher level of functionality for the paralyzed limbs. So spinal cord injury is characterized by large unmet needs in life quality and expectancy because of the limitations in mobility that affect a whole range of human activities and cause a cascade of other potential health problems. There are direct medical costs associated with the spinal cord injury, but there are also indirect costs that correspond to lack of loss of autonomy, the need to get some specific tailored support, and these costs can be as high as $2 million per lifetime on average across all conditions, for direct medical costs in Canada, much higher in the United States, and $50,000 or more per year in indirect costs. But there are some positive news there, which is most spinal cord injuries are incomplete and some recovery can occur. Incomplete meaning that there are some connection remaining between the brain and the muscles at the other end that can be retrained, and individuals with spinal cord injury can recover at least partially their movement. 70% of injuries are incomplete and they are associated with recovery. But the recovery stays partial. So can we understand what happens during this period of time where people recover their capacity to perform some functional movements and try to potentiate this recovery, to make it more complete or faster? And this is the central element of what I'm going to present today. So I briefly introduced the concept of neuromodulation, and there are several uses of neuromodulation at this very moment that are already a clinical reality, meaning that people receive electrical neuromodulation implants in their body for a variety of reasons. Some of those are still experimental, some are established. Among those that are experimental, there are some applications for spinal cord injury. So this is an experimental application for spinal cord injury. Spinal cord stimulator, which is an authorized medical device for the treatment of pain that is implanted within the body with electrodes that are in contact with the spinal cord that can deliver pulses of electrical stimulation. This is something that 50,000 people per year receive for chronic pain. And some scientists, since like in the very first cases in the late 80s, and then a lot more throughout the years, and with some very convincing demonstrations in the last decade, have been using this to try to improve movement after spinal cord injury. So the idea is that we are stimulating below the injury. So the injury will be somewhere above the positioning of the electrode. And we stimulate below the injury to excite the spinal cord that is receiving little excitation remaining from the brain above. And with this excitation, we can try to make it more receptive to the remaining brain commands and try to use it to foster the development of a functional movement. And in the video that you see here, after several weeks of training with this device, this lady is able, for the first time, to perform a step. And then to train this complex locomotor behavior over time in order to voluntarily control movement. And at any time during this process, she's always supported by this extra excitation that the scientists have been providing with spinal cord electrical stimulation. And this is not the only example of spinal stimulation in humans. Several groups have been working on this, and including invasive and non-invasive strategies. What is interesting is also that some of these groups are trying to bring this to the market, so to commercialize new medical devices that allow a better treatment for people with spinal cord injury. But I said also that neurostimulation is something that is established, and this is the case, for instance, for the brain stimulation, which is at least 12,000 people per year receive, and it's an implant in the depth of the brain. The implant doesn't leave anything else than minor scars, and it's not visible from the outside, but it allows here to, when it's programmed by the clinician, like in this case, as you can see here in real time in the implant is turned on, stimulation is delivered in the depth of the brain, and this gentleman recovers the ability to control his hands, to make fine finger movements, as you will see in a second. So it's a very striking difference between the initial condition with essential tremor, is called this condition, with uncontrollable trembling of the hands, which make any daily life activity with hand function impossible, to a situation where he has control of the fingers. He can engage in any, basically, in any functional task that he would want, like cooking or gardening or whatever is the activity of daily life that he prefers. So for this reason, the advantage for the user can be very large, both from a health standpoint and economically, because this person can go back to productive activity, which is a gain for him and for the society. And this is why this intervention becoming more and more popular. They are invasive, but they are fast growing because of the several applications that they can have in a variety of different diseases. Now, I'm gonna go through, so what do I do in this field? I'm more on the experimental side, and I'm a new professor. I started last month, so I prefer to talk today about my postdoctoral work, which I also did in Montréal, at Université de Montréal. I've been working with Marina Martinez, who is a neuroscientist working in locomotion in animal models of spinal cord injury. And I've also collaborated with other professors, including Numa Dankos, and I'm citing these two in particular because of their work being presented in my presentation today. Numa Dankos is also a neuroscientist, and he studies cortical interactions in the primate brain and in the rat brain. And as you might have imagined by my initial question, which is what happens during recovery from spinal cord injury when the injury is incomplete, recovery happens, what are those mechanisms, and how can we take them and use them to foster recovery? And I'm giving you two examples of animal models of this. So we have a rat and a non-human primate with an incomplete spinal cord injury. And in both cases, so this is a thoracic one and this is a cervical one. In both cases, this incomplete injury causes initial deficits that are quite severe. You will see here the rat completely drags the left leg, which is on the same side of the injury. And this is 5 days after an injury. But already 2 days later, this rat has already recovered an alternate stepping from left and right, which still doesn't look like a very healthy stepping, but it's an incredible amount of change for these 2 days that pass between this condition and this condition. And if you wait just a few weeks more, the rat has developed some very nice stepping, which is almost indistinguishable if you don't know the details of rat locomotion from an intact rat. So this striking recovery, we want to go and look at the mechanism behind that. I also presented this example in the monkey. This is from an article from a Japanese group published in 2007. You see that 7 days after this injury, the monkey has lost the ability of fine finger movements that allow to grasp this piece of food. But already 1 week later, you see a completely different posture. It's still not enough to have a precise grip of this food reward. But you wait 3 months, and the monkey has recovered fine finger movement and fine pinch grasp, which is a very skilled function. There are very few animals that are able to have this specific capacity of finger control. So I've been working in the rat model that you just saw. And I've been looking with Marina Martinez at the transfer of information from the motor cortex to the leg. And we've been doing that with a cortical interface, which is an implanted series of electrodes that is positioned bidimensionally within the motor cortex. And we can use that to probe the relationship between a stimulus sent in the motor cortex and the motor output that arrives at the leg. In an intact rat, you will see a lot of blue points which represent contralateral responses of the leg. So it means you stimulate the right cortex, the left leg moves. There are also some bilateral responses, meaning that both legs move. And after the spinal cord injury for the animal that I showed before, 5 days is complete dragging of the left leg. And in this situation, all the responses that we find from the motor cortex are pure ipsilateral responses, meaning that only the right leg gets activated when stimulating the right cortex. Now, you know that connections between the motor cortex and the leg are mostly crossed, meaning that our right hemisphere mostly controls the left part of the body. So in this case, you only have ipsilateral responses remaining. But then what happens the moment in which the animal has recovered some alternate stepping, you would also see the representation of the left leg starting reappearing in the motor cortex. And 1 week later, the animal has recovered a much better posture, reduced a lot the amount of dragging, and these bilateral representations are everywhere in the motor cortex, and they keep on expanding in strength as the animal keeps on progressing through recovery. So after 4 weeks, we have a motor score of 19/20, and very large and uniform representation that are also of fine ankle movements in the right cortex with respect to the left leg. So something has radically changed from here to here in the representation of the left leg during recovery, and something has radically changed in locomotor recovery. And the two elements go really well in parallel. So can we exploit this to try to potentiate this recovery? So what we just learned, can it be part of an intervention strategy? And this is what we tried to design. We try to stimulate the motor cortex during movement training in a way to try to engage more and more all the circuit from the brain to the muscle. We want to stimulate not just like any time or no matter how, we want to stimulate at the right time and at the right place. At the right place in the motor cortex that gives a very clear movement that is, for instance, a flexion of the leg, and at the right time when that flexion of the leg is required for movement. So as the rat is attempting to walk, at a certain point it's time to have a flexion of the leg, and we stimulate in the motor cortex and we help the rat supplement this flexion command that travels down to the spinal cord. Because the injury is incomplete and because we have some communication remaining and the rat is developing this communication, we are boosting this up, we're upregulating this process, and we might have, as you will see, direct effects over movement generation. So this the study that I'm going to present here, we published last year in Science Translational Medicine, where the study is much larger than the take-home message that I'm giving you. So if you're interested in this, you can go and dig deeper in all these experiments. This is a rat 7 days after injury with a clear foot drop and dragging of the left leg. And as soon as we turn stimulation on, we instantly increase this flexion of the leg. And the flexion goes up, the posture goes up, and the rat walks immediately better. I'm going to play this video again to not just to appreciate the difference between a low posture and a lot of dragging and the immediate change that happens when we turn stimulation on, but also to show you when stimulation was on. Stimulation is represented by this little light. And we're given this stimuli only at the time in which the foot has been taken off the ground. I'm gonna play this again, and you see now. So the stimulus is at the right time and is positioned in the motor cortex in the spot where we found that we had the maximum potential to obtain a specific flexion. The effects of that are dependent on dosage, meaning the more we increase the amount of stimulation delivery, the higher the foot will go. And also the more we will reduce dragging. I'm just gonna go quickly on this one by saying that the timing is important, and this is shown, for instance, in here. This wheel represents a step cycle. Here the foot takes off the ground, here the foot gets back on the ground, and here it's staying on the ground. And we can only obtain this amelioration of walking, which is represented in blue, if we stimulate right before the lift or during the swing. We cannot obtain this when we stimulate on other phases of the step. You can also see this on dragging. In order to reduce dragging, we have to deliver the stimulus right before the foot is taken off the ground. If it's too late, the foot has already been dragging and we don't see an improvement in dragging. So it's very important that the stimulus is delivered at the right time. Now, we trained these rats weekly, daily throughout 3 weeks. So starting from week 1, where these 3 different groups all have a similar performance represented by this plot here. Every day throughout 3 weeks, from week 1 to 4, we would either train the rats on walking on a treadmill with stimulation or without stimulation, or we even leave a group of them in the cage, not engaging them in any motor training. And then all these rats are tested once per week on ladder crossing, which is a different task, a task where we didn't train the rats for that. And we're trying to see, and it's a task actually where the precise control of the foot is very important. Meaning, if you don't position well your foot, then the foot is gonna fall into the ladder. And so it's a task that involves some voluntary control. It is visual motor or motor synchronization in order to perform that correctly. So the motor cortex is important in this task. And we see that rats that have received neuroprosthetic training can engage in this task without any stimulation better than all the other groups of rats. And in particular, daily treadmill training does not seem to have a benefit to a task that requires a more voluntary capacity. So the green group that received physical training against the gray group that did not, they are quite similar in the performance in this task. This is how it looks like. So the rat on top versus the rat on bottom. So the rat on top received only treadmill training, and you can see the many mistakes that she does on this ladder crossing. Gonna show you again and compare this with a better control of the foot and a better capacity to lift the foot and position that on the next rung of the ladder for the case of a rat that has received this neuroprosthetic stimulation during training. And this is the difference that was represented in the previous plot. Here, I'm going to present how also we can use this as a way to test some fancier control strategies. So what happens when if we want to control the level of kinematics that we can obtain? On top of what you saw before, so we're stimulating the brain at the right time in order to improve movement, we fix an additional external control loop where we have a PI controller, which is a regulator that tries to determine what dosage of stimulation is necessary for the task that we're doing. And this can be used, for instance, for tasks like climbing a staircase or something where we need different levels of motor output. And in this case, we're simulating this with this green line over a screen. And we are tuning our controller so that the paw of the rat tries to match as as well as possible the green line. So you see that in most cases, we can achieve quite a good fidelity of positioning for the paw that follows this virtual green line on the screen. And this is by applying a regulator on top of the, so we have an immediate controllability over foot trajectories. All right, so just to give you a general outlook on what can, now that we have established this platform, what can we do with that? I'm gonna go through 3 different developments. One is a concept of distributed neuroprosthetic intervention, which is one of my favorite concepts to develop within the next few years. And it is based on the concept that the nervous system is not a localized central computation unit. It's a distributed computation when we're talking about movement in particular. The way movement is prepared and controlled is distributed along a large variety of networks from the brain to the spinal cord. And so an intervention that targets only one center might not provide the richness of control necessary for something that is as complex as a distributed neuromotor network in the human or rat body. And just with this quick figure, I'm showing as brain and spinal stimulation together hold effects that are superior to those of spinal stimulation alone and brain stimulation alone. I'm hoping with this graph to convince you that actually there is some potential here in putting together several sources of stimulation. Let's see if some grant reviewers will actually be convinced from this. But also we can use this as a way, so the platform that I showed you as a way to ask causal questions regarding motor control. So I quickly went through the fact of movement being controlled mostly by the contralateral cortical network. So the right cortex will control mostly the left part of the body and vice versa. So what is the extent of ipsilateral control of movement? And we use this intracortical neuroprosthesis to modulate the activity of the ipsilateral cortex and show you to which extent that contributes to motor control. This is a powerful causal paradigm. And the difference between causal rather than a correlation-based paradigm, for instance, is that actually the manipulation that we do has an immediate result that we can measure rather than seeing an effect and another effect and trying to put them in, together. So this is the strength of a neurostimulation-based paradigm to ask neuroscientific questions. And in this case, I'm going to show you that the ipsilateral cortex can control extension and flexion of the ipsilateral leg. And this is a case for extension. So I talked about this dragging deficit, but there is also some postural and extension deficits involved in 1 week after injury on the more lesioned leg. So the foot lacks support. Now, I'm just going to play this back to put your attention to the right side, where we're looking at the extension of the left leg and see how the postural improvement is due to this stronger extension that we obtain from modulating the left cortex. The lesion is on the left leg is affected, and the left cortex is contributing to improving the extension of this leg. So the left cortex has a motor control, especially over the extension of the ipsilateral leg. But it can also control flexion, and this is another demonstration here. We have foot dragging after injury in this rat, and with ipsilateral stimulation, we right away obtain the same effect that I showed you before for contralateral stimulation. In some selected cases, like we really needed to find the right spot in the ipsilateral cortex that I'm going to show you in a second, but that within some very specific spots, we do obtain an effect that is similar to the one of stimulating the other cortex. It's less intense as a result, but it's there. So the left cortex has control over flexion as well of the ipsilateral leg. Here's some quantification. If you stimulate for extension, in this case, we use the specific spot in the motor cortex, the posture goes up. You have an improvement in the extensor muscle activity. The blue line is well above the gray one, which is after injury, and gets closer to what is normal before injury, which is the dark, almost black line here. You have an increase in extensor activation, but also there is some flexion capacity where you reduce dragging, you increase step height in these animals. And another example here showing how that some specific spots in the motor cortex, which is represented over here over the medial aspects, they are definitely contributing more to the left leg than the right one. As you can see over here, the response that you obtain by stimulating in this point over the left leg flexor are much stronger than the one on the right leg. And these are two animals and their muscle activity when you stimulate it on the left leg. Okay, so what about bringing this to human? I mean, we are developing neurotechnology. At the end, that what we write everywhere when we look for financing for this, when we ask a university to hire us, is we want to change the way people are treated after a specific neurotrauma. We want to give a solution. We want to change the life of people. What can we do to bring this to human? Among other things, we can try, for instance, to use less invasive procedures and studying what is the trade-off between invasiveness and efficacy. So here, rather than being inside the motor cortex, we just go on the surface of it. And the difference is that we have much less neuroinflammation and scar tissue formation, which are represented, the scar tissue formation is in red, and so the red is a marker for that, and green is a marker for neuroinflammation. When you have an implanted electrode, you will see that all around you have scars and you have neuroinflammation. And when the electrode is just on the surface, there is much less of tissue reaction to that. So there is a clear advantage on invasiveness, but there is also some loss in efficacy, meaning that the result that I show you, this immediate improvement of movement, is much less clear when the stimulation is provided on top of the brain rather than inside the motor cortex at the right spot. And there is an overall trade-off in the efficacy. We do have some more data here showing that there might be benefits for recovery of movement, but in general, I would say that the immediate effects are better when you are directly targeting the motor cortex. So this is a trade-off to take into account when designing human strategies. And we also, with Dorothy Barthélemy, who's a professor at Université de Montréal, but and also the Rehabilitation Center Gingras-Lancy de Montréal, we try to create some protocol that can let us do the things that we saw in the rat model. Would they reproduce in human if we were to have an invasive implant and try the same thing? Can we get this information without actually having an invasive implant? And we chose to do that with TMS, transcranial magnetic stimulation, which is a coil positioned on the motor cortex, and we send a magnetic pulse, we activate the motor cortex, and do we see changes in the way this individual here moves the leg when stimulation is delivered? In short, the answer is yes. Because this method is not very specific, it's not invasive. Like from the outside, the activation is not extremely precise. The effects are small, but they are very consistent. So we have changes in trajectory of about 1 centimeter but they're very consistent with the results we had in animals, meaning that the timing of stimulation is fundamental. Here you see how different timing of stimulation would have a different effect in the way the leg movement is modulated. But the overall effect, lifting the leg up, and the timing properties, the relationship with the amplitude of stimulation, everything seems to match what we saw in the rat model. And finally, the last aspect of what I'm presenting today is how to optimize neurostimulation. And I would invite you, I think this video is actually recorded on YouTube, so you will have access to these links that I'm presenting here. I would invite you to have a look at this work if you want to use the technique techniques that I'm presenting today, because I have an application all throughout neuromodulation. And what we're trying to do here is to find the relationship between a stimulation input that has so many parameters that you could select, like what's the position of that stimulus within the brain, which is three-dimensional. You might have a bidimensional grid. Well, what is the exact position of that stimulus? What's the duration, the frequency, how much current are you delivering? How long is each one of your pulse? What is the exact timing of that stimulus? So all of these parameters make selecting an optimal stimulation method quite complicated. There might require several sessions to understand what are the best ways to deliver a specific stimulation for a certain outcome that you would want. And we call this a black box relationship between these stimulation inputs and, in our case, movement output. But this output can be sensory, can be autonomic, like what's the heartbeat caused by vagus nerve stimulation, for instance. The importance is that you have something to measure, and that something is you apply a value function of that, meaning that you want to maximize or minimize that something that you're measuring. And we're using Bayesian optimization to solve this problem. So Bayesian optimization sets an explicit trade-off between exploitation and exploration. I'm gonna go for this. So exploitation is the ability to find even suboptimal solution, but quite fast. And you want to stick to them. So it's like exploitation is how good is right now your stimulation while you're still searching. And exploration is the capacity to perhaps take more time to explore, but to find the global optima on over all the parameters that you have for your stimulation. So I'll get back to here. We want to do this because it applies to many, many applications in human. I presented deep brain stimulation, spinal stimulation, but there is an entire variety of non-invasive techniques that I use currently in our rehabilitation centers, and they're kept simple because the therapists cannot handle the complexity of so many parameters. And this might be a suboptimal way of performing stimulation for rehabilitation. On top of that, new technologies present thousands of active sites. We want fully integrated, large-scale probes to perform neuromodulation, perhaps. And what's the scalability with respect to a future where you will have to test 60,000 different channels in order to find the best one for the stimulation you are delivering? This is not something that a human user could perform without a framework to optimize stimulation delivery. And so our concept is very simple here. We want to support the clinical decision or the neuroscientist decision of what stimulation to deliver with a model of the way stimulation is affecting on specific biomarkers that we are collecting. So in the case of Parkinson's disease, we might talk about deep brain stimulation and tremor or rigidity biomarkers. We put everything in a model, and the model suggests new solutions and iterates with the clinician the decision to what are the best parameters for that specific user. So this is what we're going to go through. Here the user is a rat, and we want to optimize the stimulation in the motor cortex in order to maximize the motor output. And if we lower the search space to just 2 dimensions in order to make it visible on paper, we have this scenario over here. So there are 2 dimensions, and this is maybe one location in the motor cortex of the rat, and we need to find which location is best to produce a certain movement. What we don't know is that here in this point, we have the best. So there's an initial lack of information, and the uncertainty is complete. We have a first query, and this doesn't give you much. Like, all it does, like, the uncertainty in this specific spot over here is that there is not much going on. And as we start searching, in this case, we were a little bit unlucky. We still have no particular, we have found no particular spot, but we are reducing uncertainty over time. And at a certain point, we get lucky and we stimulate in the right spot. So for the first time, we have an indication that in this area, it might be the best possible zone to stimulate. So what this algorithm goes on doing is to intensify locally and finally adopt a solution with some sporadic tests to make sure that this is the global optimum and not just like a suboptimal location. And converging on, okay, this is the ideal spot to deliver stimulation. What is also interesting of this approach is that you can exchange knowledge with the user, the human user, because you can actually inject information that will start from something that is not completely blank, but with some previous information. And at the end of the procedure, you can actually extract this and use it for your evaluation, or to average that across multiple individuals to determine what are the properties of the stimulation. So it's not like a neural network, for instance, that is quite unreadable in terms of the final weights that were found by the training. This, first of all, doesn't require any pre-training, but as it trains, all is completely transparent and can be extracted. And I'm going to show you an example here of real-time work of this optimization. So this is the same system I showed you before, but at every step, we want to use the control algorithm to select in which specific spot in the motor cortex we are going to stimulate. And this is the example. So this rat has foot drop, dragging the foot forward during stepping, and we use cortical stimulation. We want to maximize foot elevation. We can track the elevation in real time, as you can see over here. So at every step, we knew how much did the rat lift the foot. And so if we want to remove foot drop, we want to lift the foot as high as possible. But where to stimulate? This is a bidimensional representation of this rat's cortex, and we have several spots that we could use. So where within this map are we going to stimulate? And the algorithm will find out for us. So at every step, the algorithm is testing a new location in this space. And some of these tests might be not very effective, but in some other cases, as you might have just glimpsed, there are effective stimulation spots. And right now, the algorithm seems to have converged over a specific location that is quite effective, as you can see by the comparison from the before optimization and after optimization. After optimization, we completely removed this foot dragging. Okay, so, and all of this happened in real time because the learning procedure with no pre-training, the learning procedure happens iteratively in real time. You can apply this to several other applications. But before closing, I just wanted to show you something like some further applications that I'm working on right now of cortical neuroprosthetics for movement recovery. We have talked a lot about walking, but what about other functions like grasping objects? And this is a rat with an incomplete cervical spinal cord injury and lost the ability to grasp these pellets from 75% success before injury to 28% after injury. You can see there's quite some deficits in extending the hand and the arm in order to get access to that specific pellet. Now we turn stimulation on, and stimulation immediately helps the rat to better open the hand and collect the pellets. By no means this is perfect. The rat will miss quite a lot of them, but performance doubled and went very much closer to the ability of a rat that has no injury, because this is a very demanding task for a rat. They do not have the same ability we have as humans with hands. All of this is not just my work, it's actually the work of another 30 people that contributed throughout the last 5 years where I was a postdoc at Université in Montréal. And with that, I think we have another 15 minutes together for your questions.
Well, thank you very much, Professor Bonizzato, for your excellent presentation. I really enjoyed it.
Thank you, appreciate it.
It's an interesting topic, and then I've been in contact with a few researchers who are doing in this, like some interesting research in this area. It's very impactful, actually, for people with impaired mobility, and then hopefully there will be a solution soon to that particular health challenge. So I encourage participants to ask their questions using the Ask a Question box. I have a couple of questions myself to ask, but I would like first to let participants ask their questions. If there are no questions from participants, then I can start asking many questions that I have. And we'll see how much. And we have only 15 minutes. So, any questions from the audience?
Just in the meantime, I just tagged our YouTube channel on the bottom of the screen. So we gradually upload the recordings of the talk. So far, we uploaded a lot of the recording based on the permission that we have from our speakers. So again, if you want to get the update of the recording, you can just always subscribe. So on the bottom of the screen, there is a green bar that you can, click on it, and then it guides you to the YouTube channel. That's it. I just wanted to add this.
Wonderful. Well, thank you. I'm already subscribed, Matt. So my question is about these implants that you typically place in the brain or in the central nervous system. So how long do you envision these implants to be placed in this or implanted in the body? Is it like a permanent kind of implant or temporary?
Yeah. So the application can vary. For instance, in individuals with Parkinson's disease that receive this an implant in the depth of their brain would have these people have been through pharmaceutical intervention and started being resistant, for instance, to the pharmaceutical, it doesn't cover just well enough the deficits, and they then move on to a next level, which is surgical implantation. And so the implant will follow them for life, because without that, they will lose the ability to perform. Like they would have an excessive rigidity and a lot of tremor. They will need stimulation basically forever. The advantage of stimulation with respect to recordings, because you can use these interfaces for two things, reading and writing. You can read what's the activity of the brain, and we can stimulate. Reading is something that goes faster, goes away faster with time. Like the quality deteriorates quite fast, and this is an open challenge. And one of the reasons people in material study neural interfaces is that can we actually make recordings better over time? But in case of the stimulation, you would have still good capacity to inject electricity over a longer period of time. And electricity doesn't seem to be a key point in loss of biocompatibility, the injecting stimulation. So it is an advantage of neurostimulation with respect to recording for the same interfaces is that durability is much longer. But with time, still, people will need an increase in dosage. And at a certain point, it might be a very high dosage and no longer potentially safe. And that is a problematic. So there is a length of time in which you can apply within the same interface before actually having to change it, for instance, which I don't even know if it's something possible surgically. For spinal cord stimulation, probably yes. Now, in the case of what I showed you in my presentation, we are talking about fostering recovery from neurotrauma. And in that case, there is a very important phase, which is about, as soon as the immediate physical trauma is gone from maybe 1 year after a spinal cord injury or 2, that's where people get most of the recovery. This is not true for everyone, but most of the people get most of their recovery within the first year or 2 years after injury. And that is the time in which we might want to harvest as much as possible neuroplasticity in order to improve to the maximum the amount of return of function. So for cases like this, when there is no degeneration, or there is, like in Parkinson's disease, but there's a trauma and recovery, then the application is much shorter. Right? The question is really, does that individual stay for a lifetime with an implant, or is it for a certain amount of time? And I think applications where the critical time is limited, and then after that is the best that we could do, you recover to the best that we could, that's probably a more likely to be feasible from the interface perspective. So
Wonderful. So my question is now, I have a follow-up question. So it's from the material perspective, where you implant these electronic devices into the body for a long time. 2 years is also, for us at least, who are working with degradable materials, is fairly long. And when a material with a rigidity that is a bit different than the rigidity of soft tissue, such as the spinal cord tissue or the brain, when they interact, then there will be challenges with neuroinflammation and challenges of that can be detrimental to some extent. So, can you comment on the material choice for these types of electronic devices that you would like to implant in the body for, let's say, 2 years?
So historically, the material choice has been very important for neural engineering working on the domain. For a very large variety of reasons. And also, for neural interfaces used for recording or for stimulation, things change quite a lot, not just because of the specific, the electrical interface between the metal and the tissue needs to be of a different size, but also the material might be different in order to allow for different impedance properties, but also for a better capacity of stimulation. So the charge capacity is a particularly important property of the material. And for stimulation, you might want bigger sites and a material where stimulation will not cause any detrimental interaction with the tissue. Some materials are only good for recordings, like tungsten wires. They're good for recording, but they could poison the brain if you use them for stimulation, which is, by the way, what we used in the rat experiments, because the effect is not that short term. If there's 2 months in which we work with the rat, and also there was no particular, it was just, it's a cheaper way, but we will not be able to do anything similar with this specific wires in human. So usually platinum iridium, iridium oxide are popular choices for stimulation. stainless steel. For recording, you might have different, like pure platinum, because pure platinum also allows you to have quite some flexibilities in the wires or whatever you implanted. One important thing regarding the material is also the flexibility with respect to the brain. If you block your implant with respect to the skull, the brain moves within the skull, and this creates a friction between the specific wire or whatever you have and the brain. And as the two move together, you can create damage if your electrode is stiff. And this is why many companies are going for thinner and thinner electrodes. And there are some really great examples of solving this with basically wires that are so difficult to implant that you need to use a different, you basically have a stiff part to implant them, and then you remove that, and the wire is completely flexible. And we found also in our biocompatibility experiments that the material you're using, mostly in our case, was important for the compliance with respect to these movements and then the amount of damage that you would create. So all of this matter. In human, I have to say, though, we still use very large electrodes, and the material is just selected for the electrical charge transfer properties. And for what we see when it's biocompatible, no matter how long you deliver charge during the day, you would have the same amount of neuroinflammation and scar tissue formation. We did these experiments of biocompatibility within the same electrodes. So, and other groups have similar results. So I think what is mostly important when thinking of the material is the durability. So how much you will lose over time, which I don't have real good data for in the case of what I presented, and the biocompatibility of the material you're using, but also the stiffness of it and how much is compliant with the brain tissue. So these are the three key elements.
I think I'm muted. So thank you. I have many other questions that I'd like to ask, but I think given the time that is like 2 minutes time that is left, I think it's best to conclude this session. I will be in touch with you for sure after this meeting, and then we'll talk about this. But very interesting talk. Thank you very much. I would also like to thank everyone for participating in this event, and then we will see you in a few weeks again. Take care.
Perfect.
Thank you so much, Marco. I will see you, and I'll ask my question later. Take care.
Perfect. Thank you very much for the invitation. It's been a lot of fun, the discussion today, and looking forward to continue that on another occasion.
Of course. Thank you, Mosa. Bye-bye. Take care. Thank you, Marco. Bye.
Bye.